7 miles down, where pressure would crush a submarine into scrap metal, Chinese scientists just found something impossible. Thousands of creatures, thriving in absolute darkness. No sunlight, no oxygen from plants, just chemicals seeping through cracks in the earth, feeding entire cities of life that science said should not exist. A briefcase-sized drone, 33 descents, 7,000 new species, and a discovery that changes everything we thought we knew about where life can survive. If you enjoy what comes next, hit that like button and subscribe. Take a breath. We are going down. A machine no larger than a carry-on suitcase just accomplished what billion-dollar submarines cannot. Chinese engineers at Beihang University designed a transformer-like robot that swims, glides, and crawls across the deepest point on Earth. The Mariana Trench, nearly 7 miles straight down, where American Navy vessels max out at 4 miles and turn back. This tiny drone uses smart metal actuators that do something remarkable. They convert extreme water pressure into propulsion. The crushing force that destroys most machines actually powers this one. At 6.5 miles deep, the pressure equals 1,000 atmospheres. Imagine an iceberg balanced on top of a robot. That is what this machine endures every second. Professor Wen Li, the chief researcher, confirmed the drone reached depths of 35,000 feet. It did not just survive, it worked. The robotic arm collected samples, the camera recorded footage. The propulsion system maneuvered through pitch-black water, while bearing the weight of the entire ocean above it. Here's what makes this significant. In 2024 alone, China completed 246 deep-sea dives, more than every other nation on Earth combined. The United States relies on Alvin, a submersible built 61 years ago that cannot descend past 21,000 feet. The gap between Chinese deep-sea technology and Western capabilities is widening fast. The drone swam into territory that remained unexplored for decades. Each movement through the trench floor revealed sediments untouched since the planet formed. Every sample brought back contained organisms science had never documented. The machine operated where bones dissolve and metal crumples like paper. But the drone itself represents only part of the story. It is what the drone found down there that changes everything we understand about life. The robot sent back images that stunned marine biologists, not barren seafloor, not empty darkness. Instead, the camera captured something impossible. Communities, entire ecosystems, creatures clustered together in numbers that should not exist at such depths. The footage showed thousands of organisms thriving where textbooks said complex life could not survive. Scientists expected scattered microbes, maybe a few isolated single-celled organisms clinging to existence. What they discovered was a metropolis, worms, mollusks, shrimp-like creatures, all living together in dense populations, all feeding off something that is not sunlight. The drone sensors detected chemical signatures coming from cracks in the ocean floor: methane, hydrogen sulfide, carbon dioxide, poisonous compounds leaking through geological fractures. Yet instead of a dead zone, these vents created oases. The chemicals that kill most life on the planet were sustaining entire food webs 7 miles down. Every previous expedition to the Mariana Trench brought back limited data, brief visits, small samples, fragmented pictures of what exists below. China's miniature drone changed that equation. It stayed longer, traveled farther, collected more. The smart metal technology allowed it to explore areas that remained hidden from every previous mission. This tiny machine swam through valleys deeper than Mount Everest is tall. It photographed creatures that evolved in total isolation from the rest of Earth's biosphere. It documented ecosystems that operate on principles completely different from everything we see above. The drone represented a technological breakthrough. What it discovered down there represents a biological revolution. Because if life can thrive in conditions this extreme, in darkness this complete, under pressure this crushing, then our understanding of where life can exist is far too limited. And what lurks deeper still might be even stranger than anything this robot filmed. China did not send one drone down once, they sent teams, repeatedly, systematically, with the determination that redefined deep-sea exploration. Between October and December of 2021, Chinese scientists conducted an expedition unlike anything attempted before. They used Fenduzhe, China's first 10,000-meter class human-occupied vehicle. This submersible can carry actual people to the deepest zones on Earth. Not robots, not cameras on cables, living scientists who can observe, decide, and collect samples in real time. The expedition covered 3 locations: the Mariana Trench, the Yap Trench, the Philippine Basin, all Hadal zones, meaning water depths exceeding 6,000 meters. These are the places where the ocean floor drops beyond normal submarine reach, where pressure climbs so high that specialized equipment is the only option. 21 scientists joined the voyage. 17 of them descended into the abyss themselves. They rode Fenduzhe down through miles of darkness, watching the depth gauge climb to numbers most humans will never see: 11,000 meters, 36,000 feet. Deeper than commercial aircraft fly high. The numbers tell part of the story. More than 2,000 samples collected. Water, sediments, macroorganisms. 1,648 sediment samples alone. 622 specimens of a deep-sea shrimp species called Hirundellea gigas. 11 different categories of deep-sea fish. But statistics only reveal so much. What matters is what those samples contained. The scientists analyzed every specimen in detail. They sequenced DNA, measured chemical compositions, mapped genetic structures. They documented adaptation strategies that biology textbooks never mentioned. Each sample revealed another piece of a puzzle that scientists did not know existed. Fenduzhe achieved several global firsts. The submersible carried humans to the deepest point of the Yap Trench for the first time in history, nearly 9,000 meters down. This trench sits where multiple ocean currents converge. The water movement there affects weather patterns and climate across entire continents. Understanding what lives in that trench matters for understanding Earth's climate system. The expedition also marked the first systematic study of Hadal zone ecosystems. Previous missions grabbed samples and left. Fenduzhe stayed. The submersible can operate on the seabed for extended periods. It has unique sampling capacity. It is currently the only human-occupied vehicle on Earth capable of conducting thorough research in the Hadal zone system. Extended seabed operation time changes everything. Scientists could observe organisms in their natural habitat, watch how they move, see what they eat, document their behavior instead of guessing based on dead specimens pulled to the surface. The crew worked in shifts. Some dove while others analyzed samples on the ship above. The moment one team surfaced with new specimens, another team prepared to descend. Continuous operation, round-the-clock research, maximizing every moment at sea. Compare this to international efforts. In 2024, China completed 246 deep-sea dives. The United States completed fewer than 30. Japan managed around 50. Every other nation combined did not reach half of China's total. This is not exploration. This is domination. The reason matters. Deep-sea research is not just about discovering new fish. The Hadal zones contain resources, rare earth minerals scattered across the ocean floor, genetic material from organisms that survive impossible conditions, chemical compounds that could revolutionize medicine and biotechnology. Nations that map these regions first gain strategic advantages that last generations. China understands this. Their expedition schedule proves it. Fenduzhe dives again soon. More trenches, more samples, more discoveries. The question is not whether China will find more impossible ecosystems. The question is what those ecosystems will teach us before anyone else learns the same lessons. Because the samples already collected revealed something that should not be possible. And that discovery comes next. The submersible's floodlights cut through absolute darkness and revealed a nightmare that turned out to be a miracle. Scientists watching the live feed saw thousands of creatures packed together on the trench floor, nearly 6 miles down, at depths where the pressure should pulverize anything with a skeleton. Calcium cannot maintain its solid form under that much force. It dissolves into solution. Bones should not exist. Yet there they were. Mollusks with shells, worms with exoskeletons, fish with actual vertebrae, swimming through water that exerts 8 tons of force on every square inch. The laws of chemistry said this was impossible. The video feed said otherwise. The expedition collected specimens from depths ranging from 19,000 feet to over 31,000 feet. Every sample defied conventional understanding of where complex life could survive. These were not microbes, not simple bacteria clinging to rocks. These were animals, multicellular organisms with specialized organs and complex behaviors. The deepest colony ever observed was discovered at 9,533 meters, over 31,000 feet below the surface. A thriving community of tube worms and bivalves living together in dense clusters. The study published in the journal Nature confirmed this as the deepest and most extensive chemosynthesis-based ecosystem known to science. Chemosynthesis. That word holds the key to understanding how life survives where it should not. But we will get to that mechanism soon. First, consider what this depth means in practical terms. If you stood at sea level and looked up, the International Space Station orbits about 250 miles above you. If you stood at the bottom of this discovery site and looked up, you would see 6 miles of water pressing down, the weight of an entire ocean concentrated into crushing force. The temperature hovers just above freezing, 1 to 4 degrees Celsius, cold enough to slow most biological processes to a crawl. Combined with the pressure and the complete absence of light, these conditions should create a sterile wasteland, dead seafloor covered in sediment. Instead, the scientists found what one researcher called a vibrant oasis in the vast desert of the deep sea. Not isolated organisms scattered across empty terrain. Dense populations, communities, ecosystems with multiple species interacting in complex food webs. Mengran Du, a marine geochemist with the Institute of Deep Sea Science and Engineering, stated that what makes the discovery groundbreaking is not just the depth. It is the astonishing abundance and diversity of life observed. Previous expeditions found hints of deep sea life. Small samples, brief glimpses. This expedition documented entire cities of organisms thriving together. The creatures cluster around specific locations. Geological fractures where the Earth's crust cracks open. These cracks leak fluids from deep underground. Hot, chemical-rich, loaded with compounds that would poison most life on Earth's surface. But down here, in the deepest zones of the ocean, those poisons become food. The organisms have adapted to process these chemicals. They have evolved systems to extract energy from substances that kill everything else. They have developed pressure-resistant proteins that function under forces that would crush human bones into powder. Each specimen brought back to the surface revealed another adaptation, another biological solution to an impossible problem, another proof that life finds ways to exist in places we assume are dead. The implications stretch far beyond marine biology. If life can thrive under these conditions on Earth, then similar life could exist in the subsurface oceans of Europa, Jupiter's frozen moon, or Enceladus, orbiting Saturn beneath a shell of ice. Anywhere liquid water exists under high pressure, life might be possible. But the real question is not whether life exists down there. The video proves it does. The real question is how these creatures survive when every physical law suggests they should not. That answer is stranger than the creatures themselves. Imagine 50 jumbo jets stacked on top of your body, Not beside you, on you, every square inch of your skin bearing the weight of 50 fully loaded Boeing 747s. That is the pressure at the bottom of the Mariana Trench. Scientists measure it precisely, 1,086 bars, 15,750 pounds per square inch, approximately 1,072 times the standard atmospheric pressure at sea level. 8 tons pressing down on every single square inch. Put a human body in that environment unprotected, and you would not sink slowly to the bottom. You would be crushed instantly. Your bones would shatter. Your organs would compress into paste. The air in your lungs would collapse into a volume smaller than a marble. Death would be instantaneous and absolute. Yet creatures not only survive down there, they thrive, they reproduce, they have never known any other existence. The pressure at these depths affects everything. Water itself behaves differently. Sound travels faster through the compressed liquid. Chemical reactions occur at different rates. Even the light from the submersible's lamps bends slightly as it passes through water under extreme compression. To understand this force, consider what happens at different depths. At 33 feet below the surface, the pressure increases by 1 atmosphere. That is the depth where recreational scuba divers start to feel their ears pop. At 330 feet, the pressure is 10 atmospheres, deep enough that nitrogen starts to behave strangely in human blood, causing narcosis. At 3,000 feet, submarines begin to creak and groan. The hull metal flexes under stress. At 6,000 feet, only specialized military submarines can operate. At 13,000 feet, the Titanic rests on the seafloor, its steel hull slowly collapsing under relentless pressure. The Mariana Trench descends to 36,000 feet, nearly 7 miles straight down, 3 times deeper than the Titanic, 5 times deeper than most nuclear submarines can dive. When the submersible Fenduge descends to these depths, the vehicle's walls are several inches thick, made from titanium alloys designed specifically to withstand compression. The viewing ports are tiny, thick, angled to distribute force evenly. Even a microscopic crack would cause catastrophic implosion. The remotely operated vehicle called Nereus learned this lesson the hard way. It imploded during a dive in the Kermadec Trench near New Zealand. Scientists on the surface detected floating debris. The pressure had crushed the entire vehicle in an instant. One moment it was operating, normally. The next moment it ceased to exist as anything other than fragments. Human bones would suffer the same fate. Calcium carbonate, the material that makes shells and skeletons, cannot maintain its crystalline structure under that much force. The pressure breaks molecular bonds. The solid material dissolves into the surrounding water. This is why scientists initially believed complex life could not exist at Hadal depths. Without bones, how can fish swim? Without shells, how can mollusks protect themselves? Without rigid structures, how can any organism larger than a microbe maintain its shape? The answer involves proteins and cellular structures that work completely differently from anything in shallow water. These deep-sea organisms have evolved pressure-resistant molecules. Their cells contain compounds that actually require high pressure to function correctly. Take these creatures to the surface and they die. The low pressure causes their proteins to unfold and stop working. They are prisoners of the deep, adapted so perfectly to crushing force that they cannot survive anywhere else. But adaptation to pressure is only the first mystery. Because these creatures still need energy. They still need food. And in a place where photosynthesis cannot occur, where does that energy come from? The answer lies in the white, snow-like substance covering the rocks. And what that substance does changes everything. The submersible's cameras focused on patches of white material coating the seafloor. At first glance, it looked like snow. Delicate, pristine, beautiful even. The researchers called them microbial mats. They are anything but delicate. These white patches are alive, billions of microorganisms packed together in dense colonies, each one microscopic. Together they form visible structures that spread across rocks and sediment like frost on a window. Except this frost exists in water temperature just above freezing, under pressure that would turn a human into liquid. The Chinese expedition documented these mats at multiple depths: 6,000 meters, 8,000 meters, 10,000 meters. The mats appeared wherever geological cracks leaked chemicals from beneath the seafloor, always near the fractures, always clustered around the vents. These are not random bacteria. They are specialized chemosynthetic microorganisms. They have evolved to process chemicals that kill most other life forms: hydrogen sulfide, methane, ammonia. Compounds that smell like rotten eggs and burning matches. Toxic gases that would choke any air-breathing creature. Down here, in the crushing darkness, these chemicals are food. The mats glow faintly under certain wavelengths of light. Not bioluminescence like jellyfish. This is different. The chemical reactions inside the bacteria produce enough energy that sensitive equipment can detect a weak emission. Life metabolizing poison and releasing photons as a byproduct. The expedition collected samples of these mats. Scientists analyzed their composition in shipboard laboratories. They discovered extraordinary diversity. Not just one species of bacteria forming these colonies, dozens, sometimes hundreds of different microbial species living together in symbiotic relationships. Each species performs a different function. Some break down methane, others process hydrogen sulfide. Still others convert carbon dioxide into organic compounds. They work together like an assembly line. The waste product of one species becomes the food for another. Nothing goes unused. The entire system operates with remarkable efficiency. The white color comes from sulfur. As the bacteria process hydrogen sulfide, they store elemental sulfur in granules inside their cells. Billions of bacteria, each containing tiny sulfur crystals, create the snow-white appearance. These granules were first discovered in the 1880s by a scientist named Sergei Winogradsky at Hot Sulphur Springs. He hypothesized that bacteria could exist solely on inorganic chemicals. For 90 years, that hypothesis remained unproven. Then, in 1981, scientist Colleen Cavanaugh found bacteria with sulfur granules inside deep-sea tube worms. The bacteria lived in the worms' tissues. The worms had no mouth, no digestive system. They survived entirely on nutrients provided by their bacterial partners. The microbial mats in the Mariana Trench follow the same principle. They create organic molecules from inorganic chemicals. They generate energy without any input from the sun. They form the foundation of an entire ecosystem that operates on completely different rules from the rest of Earth's biosphere. The Chinese researchers confirmed that these mats produce compelling evidence of active methane generation. The microbes are not just consuming methane that leaks from vents. They are manufacturing it through biological processes, taking carbon and hydrogen from their environment and assembling methane molecules. This matters because methane is a powerful greenhouse gas. Understanding how deep-sea microbes produce it could help scientists model Earth's climate. The Hadal zones might play a larger role in atmospheric chemistry than anyone realized, but the mats serve another purpose. They feed the creatures that cannot process chemicals directly. Amphipods graze on the mats like cattle in a field. Snails scrape bacteria off rocks. Fish hunt the grazers. The white snow becomes the first link in a food chain that sustains everything else. Thousands of organisms cluster around these bacterial colonies, drawn to the only source of food in an otherwise barren landscape. The mats create islands of life in a desert of darkness, and the creatures that gather there are even stranger than the bacteria themselves. The video feed showed forests of tube worms, thousands of them, clustered so densely that their bodies formed walls of flesh anchored to the rocky seafloor. Each worm extended from a tube built around itself. The tubes stood upright like organ pipes. Some reached 6 feet tall. At the top of each tube, a bright red plume waved gently in the current. These plumes are not mouths. They are gills. The worms have no digestive system at all. They are called Symbiosis. They live at depths ranging from 6,870 meters to over 11,000 meters. The pressure at those depths would instantly crush a human skull. Yet these worms grow, reproduce, thrive. The expedition discovered tube worm communities at previously unexplored vent sites. Dense aggregations clustered around cracks where fluids seeped through the ocean floor. The worms position themselves as close to the vents as possible without getting cooked by the hot water. They need the chemicals. Without them, they starve. Hydrogen sulfide pours from the vents. This is the same compound that makes rotten eggs smell terrible. It is toxic to humans in tiny concentrations. A few hundred parts per million will kill you in minutes. These worms bathe in it constantly. The red plumes contain hemoglobin, the same oxygen-carrying molecule in human blood. But tubeworm hemoglobin does double duty. It carries both oxygen and hydrogen sulfide at the same time. The worm absorbs these chemicals from the water and delivers them to bacteria living inside its body. Inside each tubeworm is an organ called the trophosome. This structure exists solely to house bacteria. Billions of chemosynthetic microbes live inside the trophosome. The worm provides them with hydrogen sulfide and oxygen. The bacteria use those chemicals to produce energy and create organic compounds. The worm absorbs those organic compounds as food. Neither organism can survive without the other. The bacteria cannot obtain hydrogen sulfide and oxygen efficiently on their own. The worm cannot digest food because it has no mouth or stomach. They evolved together, locked in a partnership that defines both their existences. The Chinese researchers found compelling evidence that these tube worms cluster around specific types of microbial mats, the white, snow-like colonies. The worms orient themselves near these mats because the bacteria in the mats produce methane, and methane attracts other chemicals the worms need. The vents themselves create remarkable chemistry. Hot fluids from deep in the Earth's crust mix with cold seawater. The temperature difference drives chemical reactions. Minerals precipitate out of solution, metals accumulate. Entire chimney structures grow from the seafloor as dissolved minerals solidify. Some vents release clear fluids, others spew black or white smoke made of suspended particles. The black smokers reach temperatures of 400 degrees Celsius. The white smokers are cooler, but contain different chemicals. Each type of vent supports different communities of organisms. The tube worms exploit the boundary zone, the place where hot vent fluid mixes with cold ocean water. Right, at that interface, the chemistry is perfect. Enough heat to drive reactions. Enough cold to prevent cooking. Enough chemicals to feed the bacteria. Enough oxygen to support metabolism. The expedition documented tube worm fields that stretched for hundreds of meters. Entire regions of the trench floor covered in these organisms, not scattered individuals, dense populations, cities of worms living tube to tube in calculated proximity to their chemical food source. The bivalves live among them. Clams that also house chemosynthetic bacteria. These mollusks bury themselves in sediment near the vents. Their shells gape open to let sulfide-rich water flow through their tissues. More symbiosis, more impossible partnerships. Every organism in this ecosystem depends on chemicals leaking from the earth. Remove the vents and everything dies. The entire community exists because tectonic plates are pulling apart and magma heats water that seeps through cracks. But the discovery that shocked scientists most was not the tube worms. It was what scientists found living inside the microbes themselves. Photosynthesis built the world we know. Plants absorb sunlight, convert carbon dioxide and water into sugar and oxygen. Every land animal depends on this process, either by eating plants directly or by eating animals that eat plants. Sunlight is the foundation of our food web. 7 miles down in the Mariana Trench, sunlight does not exist. It has never existed. No ray of light has reached those depths in billions of years, yet life flourishes. The process is called chemosynthesis. Instead of using light energy to build organic molecules, these organisms use chemical energy. They extract electrons from inorganic compounds: hydrogen sulfide, methane, ammonia, hydrogen gas, any reduced chemical works. The bacteria combine these electrons with carbon dioxide. Through a series of reactions, they build glucose and other organic compounds, the same end products as photosynthesis, but powered by chemistry instead of sunshine. Here is how it works with hydrogen sulfide. The bacteria contain enzymes that break the bonds in hydrogen sulfide molecules. This releases electrons. The bacteria use those electrons to power a process similar to the one plants use. They fix carbon dioxide into organic material. The byproduct is elemental sulfur and water. With methane, the process is different. Methanotrophic bacteria oxidize methane molecules. They consume oxygen and methane together. The reaction produces energy. That energy drives carbon fixation. The bacteria grow, multiply, produce organic compounds that leak into the surrounding water. The Chinese expedition discovered that some Hadal bacteria use the Calvin-Benson-Basham cycle. This is the same pathway plants use for carbon fixation. The key enzyme is called rubisco. It is the most abundant protein on Earth's surface. Finding it 7 miles underwater proves that life uses similar molecular machinery in radically different environments. Other bacteria use the reductive carboxylic acid cycle. This pathway is essentially a reversed Krebs cycle, the same chemical reactions that break down food in our cells, but running backward, building molecules instead of destroying them. This cycle prefers environments without oxygen. The Hadal vents provide perfect conditions. The organisms that use chemosynthesis fall into several categories. Chemoautotrophs make all their own food from chemicals. Chemoheterotrophs consume organic compounds made by other organisms, but get their energy from chemical reactions. Each has adapted to specific niches within the vent ecosystem. Some bacteria are free-living. They form mats on rocks. Others exist in symbiotic relationships, living inside tube worms, inside clams, inside snails, providing nutrients in exchange for protection and access to chemicals. The symbiotic relationships are extraordinarily specific. Each host species partners with particular bacterial species. The tubeworm bacteria belong to different groups than the clam bacteria. Each pair evolved together over millions of years, fine-tuning their partnership until both organisms became dependent on each other. The Chinese researchers documented cooperative methanogenesis. This is a process where multiple bacterial species work together to produce methane. One species breaks down organic compounds and releases hydrogen. Another species consumes that hydrogen to make methane. Neither could accomplish this alone. Together they transform the chemistry of their environment. The temperatures where this occurs reach 55 to 80 degrees Celsius, hot enough to denature most proteins. But these bacteria thrive. They contain heat-resistant enzymes, proteins that fold differently from the ones in organisms living in cold water. Molecular structures designed to maintain function under conditions that would cook most life. The chemosynthetic process produces enough energy to support complex ecosystems. Bacteria feed amphipods. Amphipods feed fish. Fish feed larger predators. The entire food web relies on chemical reactions happening at the base. No sunlight required. No plants. No photosynthesis. Just bacteria processing poison and converting it into life. This means the deepest parts of Earth's oceans run on a completely different energy source than everything above 6,000 meters. A parallel biosphere, independent, self-sustaining, hidden in darkness. And when scientists analyzed the genetic diversity of these chemosynthetic organisms, they found something that redefines marine biology entirely. The laboratory results stunned everyone. 7,564 species-level genomes of Hadal prokaryotic microorganisms, identified, catalogued, analysed, and 89.4% of them were completely unknown to science. Let that number sink in. Nearly 90%. Not variations of known species, not subspecies or regional populations. Entirely new organisms. Bacteria that science had never documented. Archaea that existed outside every published taxonomy. Professor Xiang Xiao from Shanghai Jiaotong University led the analysis. His team sequenced genetic material from the samples. They compared the DNA sequences to every database of known organisms. The vast majority matched nothing. They represented biological diversity that had remained hidden until Chinese scientists brought samples up from the deep. The total diversity rivals all known marine microorganisms combined. Every bacteria documented in shallow seas. Every microbe catalogued from coastal waters. Every organism found in mid-ocean zones. The Hadal depths contain equivalent diversity in a tiny fraction of the ocean's total volume. This changes fundamental assumptions about where microbial life exists. Scientists thought most ocean bacteria lived near the surface, where sunlight penetrates, where photosynthetic organisms produce oxygen and organic compounds. The data now shows that the deepest parts of the ocean harbour just as much diversity as the areas we have studied for centuries. The expedition analysed 1,648 sediment samples. They extracted DNA from mud and rock. They cultured organisms in specialised equipment. They sequenced genomes using the most advanced technology available. Each new genome revealed adaptation strategies, genes for pressure resistance, genes for processing unusual chemicals, genes for surviving at temperatures that kill most life, genes that code for proteins with structures science had never documented. The microbes exhibit extraordinary novelty. They possess new genes, new molecular structures, new biological functions, resources that could provide solutions to global problems. The team specifically mentioned applications in biotechnology, medicine, and energy production. Consider what this means practically. These organisms survive conditions that destroy most biological molecules. They contain enzymes that function under extreme pressure. Pharmaceutical companies could use those enzymes to manufacture drugs under conditions impossible with current technology. The pressure-resistant proteins could lead to new materials, stronger than anything engineers currently produce. The microbes also process waste efficiently. They convert toxic chemicals into harmless compounds. They extract usable energy from substances we consider garbage. Biotechnology firms could apply these pathways to environmental cleanup, turning pollution into resources, processing industrial waste into valuable products. The genetic blueprints are now publicly available. The Chinese research team uploaded all genomic data to international databases. Any scientist anywhere can access these sequences, study them, use them for research. This level of openness is unusual in competitive scientific fields. The database represents humanity's most comprehensive catalogue of Hadal microorganisms. It includes information on metabolic pathways, gene expression patterns, protein structures, evolutionary relationships, everything needed to understand how these organisms function. The discovery also revealed something unexpected about microbial evolution. The Hadal bacteria show convergent adaptation with larger animals. Separate lineages evolved similar solutions to identical problems. Fish developed pressure-resistant proteins. Bacteria developed similar proteins through completely different genetic pathways. Evolution found the same answer twice. This convergence suggests that certain adaptations are inevitable at Hadal depths. The environment is so extreme that only specific biological solutions work. Any organism that survives there must develop similar traits. The genetic paths differ, but the functional outcome matches. The implications extend beyond Earth. If Hadal zones contain this much hidden diversity, what about other extreme environments? The hydrothermal vents in the Arctic, the hypersaline lakes under Antarctic ice, the deep biosphere inside Earth's crust. Each could harbour entire branches of the tree of life that remain undocumented. The 7,000 species represent what Chinese scientists found in just a few locations. 37 Hadal trenches exist worldwide. Most have never been explored thoroughly. Each likely contains its own unique species. Adapted to local chemistry, evolved in isolation. But the microscopic organisms are only part of the ecosystem. The creatures that feed on them are even more remarkable. Before Chinese scientists descended into the Hadal zones, marine biology was incomplete. Not slightly incomplete. Fundamentally incomplete. 90% of deep ocean life remained unknown, undocumented, existing in darkness beyond our awareness. The samples brought back from depths exceeding 6,000 meters revealed an entire dimension of biodiversity that science had missed. Not individual species here and there, entire categories of organisms, complete ecosystems, food webs that function on principles different from anything in shallow water. The expedition systematically explored the 6,000 to 11,000 meter deep regions. They did not just dip down and grab samples, they mapped, catalogued, documented everything they found. The approach was comprehensive, methodical, designed to capture complete pictures of Hadal ecology. Previous expeditions brought back fragments, a fish here, some sediment there, isolated data points that gave glimpses of deep-sea life without revealing the full story. Chinese scientists changed the methodology. They spent extended time on the seafloor, collected thousands of samples, observed organisms in their natural environment. The results showed that Hadal zones contained complex ecological networks, not scattered individuals barely surviving, thriving communities with specialized roles, predators, prey, scavengers, decomposers. Every functional group you find in a terrestrial ecosystem exists down there in adapted forms. The amphipod data alone is remarkable. 622 specimens of Hirundellea gigas collected. This species thrives at depths from 6,800 to 11,000 meters, where pressure reaches levels that should make complex life impossible. Yet these shrimp-like creatures scavenge across the seafloor, hunt smaller organisms, reproduce successfully. Scientists sequence the complete genome of this species for the first time. The genetic analysis revealed how the amphipods withstand pressure. Special proteins in their cell membranes, molecules that remain flexible under compression, adaptations in their nervous system that allow signaling to function when molecules should be crushed together. The fish data is equally significant. 11 species of deep-sea fish examined, each one surviving at depths where calcium supposedly cannot maintain solid form. Yet, they have bones, scales, organs. They swim, hunt, avoid predators, live full lives in an environment that should kill them instantly. These fish show genetic adaptations that allow survival in extreme depths. Their muscles contain modified proteins that generate force even when compressed. Their eyes have evolved to function in total darkness. Some species produce light through bioluminescence. Others have abandoned vision entirely and hunt using other senses. The diversity extends beyond what the samples directly captured. The video footage shows organisms that escaped collection, fast-swimming fish that avoided the nets, jellies that disintegrated when touched, creatures too large or too fast or too fragile to bring to the surface intact. Each glimpse of these animals raises questions. How do they navigate in complete darkness? What do they eat when food is scarce? How often do they reproduce when finding mates in vast empty spaces seems impossible? The answers remain unknown, but the footage proves the organisms exist. The 90% figure is not an exaggeration. Before this expedition, scientists had documented a small fraction of Hadal biodiversity. The comprehensive approach revealed how much remained hidden, and this was just 3 trenches. 3 locations out of 37 Hadal zones worldwide. Multiply the Chinese discoveries by the number of unexplored trenches, and you get a sense of how much life exists beyond our knowledge. Thousands of species, maybe tens of thousands, all adapted to conditions we barely understand, all evolving in isolation from the rest of Earth's biosphere. The Hadal zones represent one of the last frontiers, not in space, not on distant planets, right here on Earth, 7 miles down, accessible if you have the technology, hidden if you lack the will to explore. China made the investment, built the submersibles, trained the crews, conducted the dives, and brought back proof that life on Earth is far more diverse than our textbooks suggest. But the individual organisms reveal even stranger stories, and the smallest of them survives conditions that defy imagination. A creature smaller than your thumb withstands pressure equivalent to 50 jumbo jets stacked on top of it, every second of its life, from birth to death. Constant crushing force that would turn your body into liquid. Hirundellea gigas, an amphipod species, shrimp-like in appearance, translucent body, large eyes despite living in darkness, delicate appendages that seem too fragile to survive even gentle handling. Yet it thrives at the bottom of the deepest trenches on Earth. The Chinese researchers collected 622 specimens of this species. They found them at depths ranging from 6,800 meters to 11,000 meters. These creatures live nowhere else. They cannot survive in shallow water. The lack of pressure would cause their cells to fail. Imagine the force they endure. At 11,000 meters, the pressure reaches approximately 16,000 pounds per square inch. For comparison, a car tire holds about 35 pounds per square inch. A professional scuba tank contains 3,000 pounds per square inch. This amphipod lives under pressure 5 times greater than the strongest scuba tank. The specimens survived the journey to the surface only because scientists use specialized collection chambers, devices that maintain pressure during ascent. Let the pressure drop too quickly and the amphipods explode. Their cells rupture, proteins unfold. The delicate biochemistry that keeps them alive breaks down instantly. The genome sequencing revealed how they survive. Their cell membranes contain unusual lipids. These fat molecules remain flexible under compression. Normal cell membranes become rigid at Hadal pressures. The molecules pack too tightly together. They cannot flow. The cell cannot function. But these specialized lipids maintain fluidity even when squeezed by tons of force. The amphipods also produce unique proteins, molecules with extra bonds between amino acids. These bonds act like internal bracing. The protein folds into its functional shape and stays there even when pressure tries to collapse it. Remove the pressure and the protein unfolds slightly. The extra bonds no longer serve a purpose. The enzyme stops working correctly. Their muscles contain modified actin and myosin, the same proteins that power muscle contraction in all animals. But the Hadal versions have subtle differences. Amino acid substitutions that allow the molecules to slide past each other even when compressed. These changes are small, just a few positions in the protein sequence, but they make the difference between swimming and being paralyzed. The nervous system shows similar adaptations. Neurotransmitters function differently under high pressure. The molecules that carry signals between nerve cells change shape when compressed. In most organisms, this would disrupt brain function. The amphipods have modified neurotransmitter receptors, proteins that bind signals correctly regardless of pressure. But perhaps most remarkable is their metabolism. These creatures are scavengers. They eat whatever organic material falls from above. Dead fish, whale carcasses, any food source that sinks to the bottom. In the Hadal zones, food is rare. Months might pass between meals. The amphipods have adapted to feast or famine existence. When food arrives, they gorge. They consume as much as possible as quickly as possible. Then they enter a low energy state. Their metabolism slows. They move less, conserve energy, wait for the next meal. Their digestive system processes food with remarkable efficiency. They extract nearly every calorie, break down compounds that other organisms cannot digest. Their gut bacteria help. Microbes living inside the amphipod produce enzymes that split complex molecules. The partnership allows the amphipod to survive on sparse resources. These adaptations did not evolve overnight. The amphipods have been perfecting their pressure resistance for millions of years. Each generation slightly better adapted than the last. Natural selection operating in an environment where anything less than perfect pressure resistance means death. And they are not the only creatures that evolve solutions to impossible physics. The comparisons with what lives above get even more shocking. Take Mount Everest, the highest point on Earth, 29,029 feet tall, 8,848 meters of rock and ice reaching into the sky. Drop it into the Mariana Trench. The peak would still be 1 mile underwater. The Mariana Trench descends to nearly 11,000 meters at its deepest point. Mount Everest reaches 8,848 meters high. The difference is over 2,000 meters. More than a mile of water would cover the summit. This comparison reveals the scale of Hadal exploration. Climbing Everest requires months of preparation, specialized equipment, gradual acclimatization to thin air, teams of support climbers, weather windows, careful planning. And despite all that, the climb is dangerous. Hundreds have died attempting it. More than 7,000 people have summited Everest. The mountain sees hundreds of climbers every year during the brief seasons when conditions allow ascent. It is difficult, expensive, risky, but achievable with proper preparation. Fewer than 30 people have descended to the bottom of the Mariana Trench. Not 30 per year, 30 total. In all of human history. The first expedition happened in 1960. Jacques Picard and Navy Lieutenant Don Walsh in the bathyscaphe Trieste. They spent 20 minutes at the bottom, saw a flatfish swimming in the sediment, or thought they did. Some scientists now believe it was a sea cucumber. The next visit did not occur until 2012. 52 years later, James Cameron descended solo in a submersible he helped design. He collected samples, filmed footage, spent several hours exploring. His equipment struggled under the pressure. Batteries drained faster than expected. Hydraulic systems leaked. Thrusters malfunctioned. Victor Vescovo completed multiple dives between 2019 and 2021. He reached the deepest point several times, collected extensive samples, mapped portions of the seafloor. His submersible was specially built to withstand the pressure and operate reliably at full ocean depth. Chinese scientists have now joined this exclusive group. The Fendouzhe submersible carried 17 researchers to Hadal depths. Some descended multiple times. They collected thousands of samples, spent extended periods observing the seafloor, achieved systematic research that previous missions could not accomplish. The comparison between Everest and the Mariana Trench goes beyond depth and height. It reveals human psychology. We are drawn to heights. We can see mountains, understand them, visualize the challenge. The summit represents a clear goal. Reach the top, plant a flag, take a photo, descend. The deepest ocean is invisible, hidden beneath miles of water, dark, inaccessible. You cannot see the bottom from the surface. You cannot even see it from a few hundred feet down. The ocean swallows light. By the time you descend past 1,000 feet, the world has gone completely black. There is no flag to plant at the bottom of the Mariana Trench. No photo opportunity with a visible landmark. Just seafloor stretching in all directions. Sediment. Rocks. Maybe some organisms, if you are lucky. The achievement is abstract. You know you reached the bottom because instruments tell you so. The pressure also reverses the challenge. On Everest, climbers struggle against thinning air. Their bodies crave oxygen that is not there. At the bottom of the Mariana Trench, pressure tries to force itself into every space. The submersible walls keep it out, but you feel the compression. Hear the metal creaking. Know that mere inches of titanium separate you from instant death. The ocean is Earth's last frontier. We have better maps of Mars than we do of the deep sea floor. We have sent more people to space than to Hadal depths. The cost, the danger, the technological challenges, all conspire to keep these places unexplored, until China decided that understanding what lives down there is worth the investment. And what they found suggests we should have started exploring decades ago. Every ecosystem needs energy. In forests, sunlight powers photosynthesis. Plants grow, herbivores eat plants, carnivores eat herbivores. Energy flows upward through trophic levels. Remove the sun and the forest dies. 7 miles down in the Mariana Trench, no sunlight has ever penetrated. Yet a complete food web functions. Multiple trophic levels. Energy flowing from primary producers to apex predators. A self-sustaining ecosystem that operates independently of everything above. The Chinese researchers mapped this food web for the first time. They identified each component, documented the connections, traced energy flow from chemicals to microbes to animals. The resulting picture shows complexity rivaling any terrestrial ecosystem. The base layer is chemosynthetic bacteria. They fix carbon dioxide into organic compounds using energy from chemical reactions. These microbes are the primary producers. Everything else depends on them. They form mats on rocks, live inside tube worms, partner with clams, grow in sediments. The first consumers are grazers. Amphipods scrape bacteria off rocks. Snails consume microbial mats. Small worms filter bacteria from sediment. These organisms convert microbial biomass into their own tissues. They grow, reproduce, become food for the next level. Mid-level predators hunt the grazers, fish species that have evolved to live at Hadal depths. They detect prey using lateral lines that sense vibration. In total darkness, vision is useless, but water movement reveals location. A swimming amphipod creates pressure waves. The fish feels them, strikes with precision. Some fish produce bioluminescence. They have light-generating organs that flash in patterns. The purpose is debated. Possibly communication, maybe prey attraction, perhaps mate recognition. In an environment where finding another member of your species is nearly impossible, any signal helps. Scavengers occupy another niche. They wait for organic material to fall from above. When a whale dies at the surface, its body eventually sinks. The carcass settles on the Hadal floor. Scavengers converge. Amphipods in the thousands, fish, worms, anything mobile enough to reach the food source. A single whale fall can sustain a local ecosystem for years. The scavengers pick the soft tissue. What remains becomes a structure. Bones provide habitat. Bacterial colonies form on the skeleton. Specialized worms bore into the bones. Extract lipids still present in the marrow. Even the skeleton becomes food. The Chinese expedition found evidence of multiple food sources, not just whale falls, dead fish that sink from shallow depths, organic particles that rain down constantly, algae and plankton that grow at the surface and die. Their bodies descend through the water column. By the time they reach Hadal depths, they have been picked over repeatedly. But some material remains, enough to supplement the diet of deep-sea organisms. The food web also includes decomposers, bacteria that break down dead organisms, fungi that process organic debris. These organisms recycle nutrients. They convert complex molecules back into simple chemicals. Those chemicals feed the chemosynthetic bacteria. The cycle completes. This closed-loop system is remarkably efficient. Almost nothing is wasted. Every organism fills a role. Every waste product becomes a resource for something else. The ecosystem operates with minimal input from above and sustains itself through internal cycling. The diversity within each trophic level provides resilience. Multiple species of amphipods, several types of fish, different bacterial communities. If one species declines, others can fill the gap. The food web adapts, continues functioning even when conditions change. The Chinese researchers also documented seasonal variations. Food availability fluctuates. During certain times of year, more organic material sinks from surface waters. The Hadal organisms respond, reproduction increases, growth rates accelerate, then the input decreases, and the ecosystem enters a slower phase. But the most surprising finding was about the fish. Their adaptation to pressure involves chemistry that scientists thought they understood. Turns out everything we believed about deep-sea fish physiology was wrong. The Chinese researchers found compelling evidence that changes our understanding of deep-sea chemistry. Microbes in the Hadal zones are not just consuming methane, they are producing it, manufacturing the gas through biological processes in conditions that should make such reactions impossible. Methane is a simple molecule, one carbon atom bonded to 4 hydrogen atoms, but creating it requires specific conditions. Life makes methane through methanogenesis. Specialized archaea called methanogens perform this process. They take hydrogen and carbon dioxide and assemble methane molecules. Methanogenesis typically occurs in swamps, rice paddies, cow stomachs, environments that are warm, organic-rich, and oxygen-free. Scientists assumed deep-sea methanogenesis followed the same rules, microbes consuming organic matter and releasing methane as waste. The Hadal samples revealed something different. These methanogens operate at extreme pressure, near freezing temperature, with limited organic material, yet they produce methane at rates comparable to shallow water methanogens. They have adapted their biochemistry to function under conditions that should halt the reactions. The key is cooperative methanogenesis, multiple species working together. One archaeal species breaks down what little organic matter exists. This releases hydrogen gas. Another species consumes that hydrogen to make methane. Neither could accomplish the full process alone. Together they create an assembly line. The temperatures at the vent sites range from 55 to 80 degrees Celsius, hot by deep-sea standards, but the methanogens live in the surrounding sediments where temperatures drop to just above freezing. They process hydrogen released by the thermophilic organisms near the vents. This spatial separation allows both warm-adapted and cold-adapted microbes to coexist. The methane production matters for several reasons. First, it provides an additional energy source for the ecosystem. Methanotrophic bacteria consume the methane. These organisms oxidize methane using oxygen dissolved in seawater. The reaction produces energy they use to fix carbon dioxide. More primary production, more food for the ecosystem. Second, methane is a greenhouse gas, 25 times more potent than carbon dioxide at trapping heat. Understanding where it comes from matters for climate science. If Hadal zones produce significant methane, and that methane reaches the atmosphere, it affects global temperatures. Scientists are still determining how much methane escapes the deep ocean. Most gets consumed by bacteria before reaching the surface, but some makes it through. Methane bubbles rising from deep-sea vents have been documented. The question is quantity. How much it reaches the atmosphere versus how much gets oxidized in the water column. The Chinese researchers also found evidence of hydrogen-based chemosynthesis. Some organisms use hydrogen gas directly. They oxidize molecular hydrogen and use the energy to fix carbon. This pathway is less common than sulfide-based or methane-based chemosynthesis, but it appears in environments where hydrogen is abundant. The Hadal vents release hydrogen through geological processes. When seawater reacts with certain minerals in the Earth's crust, hydrogen gas is produced. This process is called serpentinization. It occurs when water interacts with iron-rich rocks. The chemical reaction releases hydrogen and heat. Organisms that use hydrogen as an energy source have a nearly unlimited supply. As long as the geological processes continue, hydrogen keeps leaking from the vents. The microbes harvest it, convert it into biological energy, support themselves and everything that feeds on them. The discovery of active microbial methane production at Hadal depths suggests similar processes could occur in other high-pressure environments. The subsurface ocean on Europa, Jupiter's ice-covered moon, experiences pressures similar to Earth's Hadal zones. If life exists there, it might use methane metabolism, just like the organisms in the Mariana Trench. The microbes also show remarkable genetic flexibility. Their genomes contain multiple metabolic pathways. When one chemical becomes scarce, they switch to another. This adaptability allows survival when environmental conditions change. Vent chemistry fluctuates, flow rates vary. The microbes adjust their metabolism accordingly. But while microbes show incredible adaptation to pressure, the fish reveals something even more unexpected. A discovery that overturned decades of established science. For decades, scientists believed they understood how fish survive at extreme depths. The theory was elegant, simple, supported by data from multiple studies, and completely incorrect. The theory centered on a compound called trimethylamine N oxide. Scientists refer to it as TMAO. This organic molecule appears in fish tissues. It helps stabilize proteins under pressure, acts like a molecular brace, keeps enzymes folded correctly when tons of force try to crush them. Research showed that TMAO concentrations increase with depth. Shallow water fish have low levels. Mid-depth fish have more. Deep-sea fish have high concentrations. The correlation was clear. As pressure increases, fish produce more TMAO to protect their proteins. The pattern made biological sense. Pressure denatures proteins, unfolds them, disrupts the precise three-dimensional shapes that allow enzymes to function. TMAO counteracts this effect. The molecule inserts itself between protein regions, provides structural support, prevents collapse. Scientists extrapolated. If the pattern continues, then Hadal fish should have extremely high TMAO concentrations, maybe double or triple the levels found in mid-depth species. This became the accepted model. Textbooks included it. Research papers cited it. Everyone believed it. The Chinese expedition tested the theory. They collected 11 species of deep-sea fish, analyzed tissue samples, measured TMAO concentrations. The results shocked the research community. Below 6,000 meters, TMAO levels not increase. In some species, they actually decrease. The Hadal fish survive crushing pressure with TMAO concentrations lower than expected, based on the established gradient. The correlation breaks down precisely where it should be strongest. This finding challenges fundamental assumptions about deep-sea adaptation. If TMAO is not the mechanism protecting Hadal fish proteins, then what is? Something else must provide pressure resistance. Some other molecular strategy that science has not identified. The researchers examined protein structures directly. They found modifications at the genetic level, amino acid substitutions that change how proteins fold, small tweaks in the sequence that dramatically alter pressure resistance. These changes do not appear in shallow water fish. They evolved specifically in Hadal lineages. The proteins in Hadal fish contain more charged amino acids, residues that form ionic bonds with each other. These bonds create additional crosslinks within the protein structure. The molecule becomes internally reinforced. Pressure cannot collapse it as easily. The fish also show changes in membrane lipids. Their cell membranes contain unsaturated fats that remain fluid under compression. This prevents membranes from becoming rigid, allows transport processes to continue, keeps cells functioning when pressure should lock everything down. But the TMAO finding raises bigger questions. If the established model was wrong for Hadal fish, might it be wrong for other depths too? Perhaps mid-depth fish use different mechanisms than scientists assumed. Maybe the TMAO correlation is coincidental rather than causative. The research team published their findings in the journal Cell. The paper presents the data clearly. TMAO concentrations in Hadal fish do not match predictions. The field must reconsider established theories. This is how science progresses, not through confirmation of existing ideas, through discoveries that contradict them. The Hadal expeditions provided data that shallow water research could never generate. Access to extreme environments reveals which theories hold up under testing. The genetic sequencing also revealed other adaptations. Hadal fish have modified visual systems. Their eyes contain proteins that function differently from surface fish. Some species have lost functional vision entirely. Others have enhanced other senses. Lateral line systems that detect minute pressure changes. Olfactory receptors that track chemical gradients in complete darkness. The fish genomes show evidence of convergent evolution. Different Hadal species evolved similar solutions independently. Pressure-resistant proteins with different sequences, but similar structures. This suggests that certain molecular architectures work better under extreme pressure. Evolution discovers these solutions repeatedly. But while fish adapted through genetic changes over millions of years, other organisms evolved even stranger strategies. And one group of creatures reveals the most impossible adaptation of all. Calcium carbonate cannot exist in solid form at Hadal pressures. The chemistry is clear, unambiguous. Under that much force, the molecular bonds break. The solid crystals dissolve, return to solution. The water becomes saturated with calcium ions and carbonate ions, but no solid material remains. This is why scientists assumed shelled organisms could not survive in Hadal zones. Shells are made of calcium carbonate. So are the skeletons of many marine invertebrates. Remove the ability to maintain solid calcium, and these organisms should cease to exist. The Chinese expedition discovered thousands of mollusks living at depths approaching 6 miles. Clams, snails, bivalves with visible shells. These organisms violate the chemical principles that govern calcium solubility. Yet there they are, collected in jars, documented in photographs, analyzed in laboratories. The shells are thinner than their shallow water relatives, more delicate, but they exist. The mollusks build them, maintain them, use them for protection and structural support. Despite chemistry that says this should be impossible, the researchers examined shell composition. They found modifications to the crystal structure. The calcium carbonate crystals are smaller, more tightly packed, reinforced with organic matrix materials. The mollusk secretes proteins that bind to the calcium. These proteins create a framework. The calcium deposits within that framework. The combination is more stable than pure calcium carbonate. The organic matrix acts like rebar in concrete. The calcium provides hardness. The proteins provide flexibility and resistance to cracking. Together they create a composite material that withstands pressure better than either component alone. But even with modifications, the shells remain vulnerable. The mollusks must constantly repair them. Pressure tries to dissolve the calcium. The mollusk adds new material. A continuous process. Stop for too long and the shell weakens, cracks, eventually fails. The energy cost is enormous. Building shell material requires calcium. In deep water, dissolved calcium concentrations are lower than at the surface. The mollusk must extract calcium from a dilute source, transport it through tissues, concentrate it, combine it with carbonate, secrete the mixture, all while maintaining metabolic processes under extreme pressure. These mollusks also house chemosynthetic bacteria, symbionts living in their gills. The bacteria process sulfide from the vents, produce organic compounds, feed the mollusk. This partnership allows the mollusk to devote energy to shell maintenance. Without the bacterial food source, the mollusk could not afford the metabolic cost. The shells also serve defensive purposes. Predators exist at Hadal depths. Fish that hunt mollusks, crabs that crack shells. The protection matters. A mollusk without a shell does not survive long. Natural. Selection strongly favors individuals that maintain shell integrity. The Chinese researchers found evidence of shell damage and repair. Growth lines showing where cracks occurred. New material deposited to seal breaches. The mollusks survive injuries that would kill shallow water species. They have adapted not just to build shells under pressure, but to repair them continuously. Some mollusks burrow into sediment, bury themselves with only their siphons exposed. This reduces predation risk. Also provides access to pore water, spaces between sediment grains where chemistry differs from the open water. Some burrowing mollusks extract chemicals from pore water to support their bacterial symbionts. The discovery of thriving mollusk populations at Hadal depths forces a reconsideration of chemical boundaries. Science said calcium carbonate shells cannot exist. Biology found a way. The organisms evolved strategies that bypass the limitations, changed the rules through adaptation. This pattern repeats throughout Hadal ecology. Every time chemistry or physics says something is impossible, evolution demonstrates otherwise. Pressure should denature proteins. Organisms make pressure-resistant proteins. Calcium should dissolve. Organisms build reinforced shells. Darkness should prevent vision. Organisms abandon eyes and develop other senses. Life does not accept boundaries. It finds solutions, adapts, survives, thrives even in conditions that seem fundamentally incompatible with complex biology. And the scope of China's research into these impossible organisms reveals strategic thinking that goes far beyond basic science. Between October and December 2021, China conducted 33 dives into the deepest parts of the ocean. Not scattered missions over years. 33 descents in 2 months, using Fendouzhe, the only human-occupied vehicle capable of systematic Hadal research. This was not exploration. This was a campaign. Each dive collected samples, documented organisms, mapped seafloor features, tested equipment. The data accumulated rapidly. By the end of the expedition, Chinese scientists had compiled the most comprehensive dataset of Hadal ecology ever assembled. More information than all previous expeditions combined. This scale of operation requires infrastructure, support ships, trained crews, specialized equipment, laboratories for processing samples, communication systems, navigation tools. The logistics alone represent a massive investment. China committed the resources, built the capacity, executed the plan. The strategic implications extend beyond biology. The Hadal zones contain resources that nations want, rare earth elements, scattered across the seafloor. These minerals are essential for modern technology. Smartphones, electric vehicles, wind turbines, military equipment. Every advanced economy depends on rare earths. Currently, China dominates global rare earth production. They control over 80% of supply. This gives them leverage in international trade. In 2020, China increased their mining quota to a record 140,000 tons. They are expanding operations in Greenland, investing in rare earth companies across multiple continents. But land-based deposits are finite. They will be exhausted eventually. The ocean floor holds vastly larger quantities. Japan discovered rare earth deposits in the Pacific. Recoverable reserves potentially 1,000 times greater than land sources. The seabed represents the next frontier for resource extraction. International law attempts to regulate deep-sea mining through the International Seabed Authority. But no final agreement exists. Nations are positioning themselves, claiming exploration rights, developing technology, testing extraction methods. When regulations are finalized, the countries with operational capabilities will have advantages. China is leading that race. Their deep-sea diving capabilities exceed all competitors. American submersibles are outdated. The Alvin is 61 years old. It cannot reach Hadal depths. Japan has capable vehicles, but conducts fewer missions. Europe collaborates on research, but lacks independent operational capacity at the scale China has achieved. The 33 dives demonstrate technical superiority. Fendouzhe performed reliably, extended bottom time, collected diverse samples, operated in challenging terrain. The missions proved Chinese engineers can build vehicles that withstand extreme conditions and function consistently. But the expeditions also provided cover for mapping, Every dive creates detailed sonar images, topographic data, sediment composition, current patterns. This information matters for future operations. If China decides to mine the Hadal zones, they already know the terrain. They have identified resource-rich areas. They understand the challenges. The biological discoveries provide additional strategic value. The genetic database contains blueprints for pressure-resistant proteins, enzymes that function in extreme conditions, metabolic pathways that process unusual chemicals. This biological intellectual property could be worth billions. Pharmaceutical companies want extremophile enzymes. They enable drug manufacturing under conditions impossible with conventional catalysts. The compounds are more stable, the reactions more efficient, the products purer. Companies will pay premium prices for access to these genetic resources. Biotechnology firms want the metabolic pathways, organisms that convert waste into valuable products, microbes that clean up pollution, bacteria that extract metals from low-grade ore. The Hadal organisms demonstrate processes that industry desperately needs. China made all the genomic data publicly available. This seems generous. Share discoveries with the world. Advance science globally. But the genetic sequences alone are not enough. You need the organisms themselves to study protein, function, test metabolic pathways, develop applications, and China has the samples. They control access to the living organisms. The research also builds scientific prestige, publications in top journals, international recognition, leadership in deep-sea biology. This establishes China as the authority on Hadal ecosystems. When policy decisions are made about deep-sea mining, Chinese scientists will be consulted. Their data will inform regulations. Their expertise will shape international agreements. The 33 dives were never just about science. They were about positioning, demonstrating capabilities, gathering intelligence, securing future advantages, and the discoveries made during those dives might hold solutions to problems far beyond the ocean. The tube worms cluster in predictable patterns, always near geological fractures, always oriented toward the cracks. Thousands of individuals positioned with mathematical precision around specific points on the seafloor. These are not random distributions. This is calculated survival. The fractures in the Earth's crust are where tectonic plates pull apart. Stress builds, rock breaks, cracks open through layers of sediment and bedrock. These openings create pathways, routes for fluids deep in the crust to travel upward, heated by magma, pressurized by overlying rock. The fluids seek release. When the fluids emerge, they carry dissolved chemicals, hydrogen sulfide, methane, carbon dioxide. Minerals precipitate as temperature drops. The surrounding seawater is cold, near freezing. The hot fluids cool rapidly. Sulfides solidify, form chimneys, create structures that can grow meters tall. The Chinese researchers documented multiple vent types at Hadal depths. Black smokers releasing dark plumes. The color comes from metal sulfides, iron, copper, zinc, all dissolved in the hot fluid. They precipitate when they contact cold water. The result looks like smoke, but is actually suspended particles. White smokers release lighter colored plumes. These vents are cooler. The chemistry is different. They emit more barium and calcium, less metal sulfide. The white appearance comes from barium sulfate crystals, smaller particles, different composition, supporting different microbial communities. Clear vents emit fluids without visible plumes. The dissolved chemicals are different, less prone to immediate precipitation. These vents can be difficult to locate visually, but organisms cluster around them just as densely as around black or white smokers. The tube worms detect the chemicals. They do not need to see the vent. Each fracture creates a microenvironment. The hot fluids mix with cold seawater at specific ratios. This creates chemical gradients. Near the vent opening, conditions are too hot, too chemically extreme. Farther away, the fluid is too diluted. Between these zones exists a sweet spot, perfect chemistry. Perfect temperature, maximum energy availability. The tube worms exploit this zone. They position their tubes at the optimal distance, close enough to access chemicals, far enough to avoid heat damage. This positioning requires sensing capabilities. The worms detect temperature, chemical concentrations, water flow. They adjust their position accordingly. The fractures also determine community composition. Different vent types support different organisms. Black smoker communities include species that tolerate high metal concentrations. White smoker communities have organisms adapted to barium. Clear vents support species that depend on methane or hydrogen rather than sulfide. The Chinese expedition mapped vent distribution across the trench floor. They found clusters, multiple vents in close proximity, then large gaps, empty seafloor where no vents exist. The organisms concentrate in the vent clusters. Between clusters, the seafloor is nearly barren. A few scavengers, some microbes in sediment, but no dense communities. This creates an archipelago effect. Islands of life, separated by deserts. Organisms in one cluster may be genetically isolated from organisms in another cluster, even if they belong to the same species. The distance prevents migration, prevents gene flow. Over time, populations diverge. The expedition also documented how long vents remain active. Some emit fluids continuously for decades. Others are episodic, active for months, then dormant, then active again. The organisms must adapt to this variability. Tube worms can survive short periods without vent flow. They store energy, slow their metabolism, wait for flow to resume. When a vent dies permanently, the community collapses. The tube worms cannot relocate. They are anchored, fixed to the seafloor. Without vent chemicals, their bacterial symbionts starve. The worms starve. The entire community disappears. New fractures open elsewhere. New vents form. New communities establish. This cycle has been happening for millions of years. Vents appear. Communities grow. Vents die. Communities collapse. The process shapes evolution, drives adaptation, creates the diversity Chinese scientists documented. But what lives in these vent cities might hold answers to questions that reach far beyond geology and biology. The colossal squid is heavier than the giant squid, shorter in total length, but more massive, more muscular, built for power instead of reach, and its tentacles are covered in weapons that spin. Most squid have suckers, circular discs that create vacuum seal. They grip prey, hold it tight. But colossal squid evolved something different, something worse. Their tentacles have hooks, sharp, curved, made of chitin. Each hook is maybe an inch long, razor-edged, and they rotate. The hooks are mounted on swivels. They can turn 360 degrees. When the tentacle wraps around prey, the hooks dig in from every angle. They rotate, cut deeper, shred flesh. This is not a grip. This is an attack. The hooks do not just hold prey. They damage it, tear it apart, make escape impossible. Colossal squid hunt in the Southern Ocean, around Antarctica, at depths up to 7,000 feet. The water is near freezing, dark, filled with fish and smaller squid. They are ambush predators. They wait in the water column, motionless. When prey passes within range, the tentacles shoot out, faster than human eyes can track. The hooks embed. The rotation begins. Prey has no defense against this. The hooks cut through scales, through skin, into muscle. The more the prey struggles, the worse the damage becomes. Colossal squid also have hooks on their arms. 8 arms, 2 tentacles, all covered in rotating weapons. When they pull prey toward their beak, every surface that touches the prey is cutting it. The beak itself is massive, parrot-like, made of chitin harder than most metals. It can bite through bone, through steel cable, through anything organic. But the beak is not the primary weapon. The hooks are. They incapacitate prey before it reaches the beak. By the time the squid begins eating, the prey is already shredded. Researchers have examined scars on sperm whales, circular marks from sucker rings, and slash marks from hooks. The hook wounds are deeper, more severe. They take longer to heal. Some sperm whales carry hook scars across their entire head, evidence of battles with colossal squid. The whale won, obviously. But the squid did not go quietly. Colossal squid can weigh over 1,000 pounds. They have the largest eyes of any animal relative to body size, even larger than giant squid eyes, almost 12 inches across. These eyes are designed to see in near darkness, to detect bioluminescence, to spot sperm whales hunting them from far away. The squid's mantle is thick, muscular, filled with ammonia-rich tissue that provides buoyancy. The ammonia makes the flesh toxic, inedible to most predators. Sperm whales do not care. They have evolved tolerance to ammonia. They can eat colossal squid without getting poisoned. They are one of the few predators capable of hunting these massive creatures. Colossal squid reproduction is mostly unknown. We have never observed them mating. We assume they spawn like other squid. Release eggs into the water. Let them develop on their own. But everything about colossal squid is assumption. We have only examined a few dozen specimens. Most were dead when found, floating at the surface, or caught in fishing nets. We have never seen a living colossal squid in its natural habitat, never watched it hunt, never observed its behavior. Everything we know comes from corpses and analysis. The rotating hooks were discovered by examining preserved tentacles under magnification. The swivel mechanism is intricate. Biological engineering at a microscopic level. Evolution created a joint that allows full rotation without tearing tissue. Each hook has muscles attached. The squid can control the rotation, can make the hooks spin or lock in place, can adjust the angle of attack. This level of control requires a sophisticated nervous system. Colossal squid brains are large, complex. They process information from millions of nerve endings in their arms and tentacles. They are intelligent, probably more intelligent than we realize, but we have no way to measure it. No way to test cognition in an animal that lives so deep and dies so quickly when brought to the surface. What we know is that evolution gave them weapons unlike anything else in the ocean. Hooks that rotate, that cut from every angle, that make escape impossible. And these weapons exist on a creature that weighs half a ton, that moves through the darkness like a ghost, that can accelerate to incredible speeds using jet propulsion. Colossal squid are apex predators in the southern ocean depths. They have few natural enemies besides sperm whales. They are at the top of their food chain, but even apex predators have limits. Even they cannot survive everywhere. There are zones below where they hunt, zones where the pressure increases even more, where the environment becomes even more hostile. At 13,000 feet, you cross into the abyssal zone, the abyss. The word comes from Greek. It means bottomless, infinite depth. The name fits. Pressure here exceeds 6,000 pounds per square inch. Temperature is constant, 35 to 39 degrees Fahrenheit. Darkness is absolute. Not a photon of sunlight reaches this depth. The abyssal zone covers more than half of Earth's total surface area. It is the largest habitat on the planet. Bigger than all the continents combined. Bigger than all forests, all deserts, all shallow oceans. And we have explored less than 1% of it. The seafloor here is not flat. It is not smooth. It is rugged. Mountains rise thousands of feet. Canyons cut thousands of feet deeper. Ridges run for hundreds of miles. Volcanic vents spew superheated water. Most of the floor is covered in sediment, fine mud made from the dissolved shells of dead organisms, particles that have been drifting down for millions of years. In some places, The sediment is thousands of feet thick. This sediment is the foundation of the abyssal ecosystem. Organisms burrow into it, feed on it, process it. They extract nutrients from particles that have been settling since before humans existed. The food web here depends entirely on what falls from above. Marine snow, dead organisms, fecal matter, whale carcasses. Everything that sinks eventually reaches the abyssal seafloor. When a whale dies at the surface and sinks, it becomes an entire ecosystem. A whale fall. The carcass lands on the seafloor and hundreds of species converge. First come the mobile scavengers, sharks, hagfish, crabs. They strip the soft tissue, the meat, the blubber. This phase lasts months. Then come the smaller scavengers, worms, snails. They pick the bones clean, extract nutrients from cartilage and bone marrow. This phase lasts years. Finally, bacteria colonize the skeleton. They break down the lipids stored in the bones. This phase lasts decades. The whale skeleton supports life for 50 years or more. For organisms adapted to starvation, a whale fall is a treasure. A concentration of nutrients that might sustain a community for generations. But whale falls are rare. Most of the abyssal zone receives only a thin drizzle of marine snow. A few grams of organic matter per square foot per year. Organisms here exist on almost nothing. Their energy budgets are smaller than any surface animal. They move slowly, grow slowly, reproduce rarely. Many abyssal fish are blind. Eyes cost energy to maintain, energy to power. If there is no light, eyes are useless. Evolution removes them. These fish navigate by touch, by chemical sensing, by detecting water movement through lateral lines. They build mental maps of their territory using senses humans barely understand. Some abyssal organisms are transparent. Their bodies are see-through. You can watch their organs work, see their digestive system, see their blood pumping. Transparency offers no camouflage here. There is no light to hide from. But it might reduce the energy cost of producing pigments. Every saved calorie matters. The abyssal zone has distinct regions. Areas near underwater mountains have different species than flat plains. Areas near volcanic vents have entirely different ecosystems. Hydrothermal vents support life that does not depend on sunlight. Chemosynthetic bacteria use chemicals from the vents as energy. They form the base of a food chain that includes tube worms, clams, and specialized fish. These vent communities exist in islands of warmth, surrounded by freezing abyssal water. The temperature gradient creates boundaries. Species adapted to vent heat cannot survive in the surrounding cold. Species from the cold water cannot survive the vent heat. The abyss is not uniform. It is a patchwork of microhabitats, each with its own conditions, its own species, its own rules. Currents flow across the abyssal plains. Slow currents, they move at inches per hour. But over years, they transport nutrients across thousands of miles. They connect distant ecosystems, carry larvae from one region to another. Some abyssal organisms spend their entire lives moving with these currents, drifting, never touching the seafloor, living in the water column above the abyss. Others are sessile, attached to the bottom, waiting for food to drift past. They might wait months, years. They have evolved patience beyond comprehension. The abyssal zone also contains most of Earth's undiscovered species. Every expedition that sends cameras or nets to these depths finds new organisms. Species never before documented. Estimates suggest we have identified less than 10% of abyssal species. The rest are unknown, unobserved, living in darkness that humans have never penetrated. And below the abyss, there is something even deeper, even more extreme. The Dumbo octopus floats above the abyssal seafloor. Its ear-like fins flap slowly, gracefully. It looks like it is flying through water. The fins are the reason for the name. They resemble the ears of Disney's Dumbo the elephant. They extend from the side of the head, flap in rhythmic pulses, provide propulsion and stability. Most octopuses crawl. They use their arms to pull themselves across the seafloor. They hide in crevices, in caves. They are bottom dwellers. Dumbo octopuses abandoned that lifestyle. They evolved to swim, to hover, to live in the water column far above the bottom. They are the deepest living octopuses on Earth, found at depths exceeding 23,000 feet, in the Hadal zone, in trenches, in places where pressure should prevent complex life. Their bodies are soft, gelatinous, almost translucent. They have minimal muscle density. This reduces their weight, lets them float without constant effort. The fins provide fine control. The octopus can hover in place, can drift with currents, can accelerate when needed using jet propulsion from its siphon. They feed on small invertebrates, copepods, amphipods, worms. They drift through the water until prey comes within range. Then the arms extend, grab, pull the prey to the mouth. The arms are webbed, connected by tissue that forms a bell shape when spread. The octopus can use this webbing like a net. It descends over prey, traps it. Then the arms bring it inward to the beak. Dumbo octopuses have relatively large eyes. Not as large as giant squid eyes, but substantial for an octopus. They can detect bioluminescence, track glowing organisms in the darkness. They also have photophores, light-producing organs scattered across their body. They can glow, flash patterns, communicate with other Dumbo octopuses. We think they use bioluminescence for mating displays. Males might flash specific patterns to attract females, but we have never observed this. Never documented Dumbo octopus courtship. Females lay eggs on the seafloor. They attach them to rocks or coral skeletons. Large eggs, well provisioned. The embryos develop slowly over months. When they hatch, the young octopuses are miniature adults. They swim immediately, begin hunting on their own. No parental care, no protection. Dumbo octopuses come in various colors. Some are pale, almost white. Others are pink, red, orange. The color might indicate species, or it might be individual variation. We do not know. Different species have been identified based on fin shape, arm length, body proportions. There might be 18 species of Dumbo octopus, maybe more. Classification is ongoing. They are rarely seen. Living at extreme depths makes observation difficult. Most of what we know comes from specimens caught in deep-sea trawls, or footage from remotely operated vehicles. The footage shows them moving with eerie grace, fins flapping, arms trailing. They look peaceful, serene, like creatures from another world. But they are effective predators. Their feeding strategy is patient. They drift through huge volumes of water. Eventually they encounter prey. The arms shoot out. The capture happens in seconds. Dumbo octopuses have relatively simple brains compared to shallow water octopuses. They are not tool users. They do not solve complex puzzles. They do not change color for camouflage. Their intelligence is focused on navigation, on hunting, on surviving in an environment with minimal sensory input. They can sense vibrations through their arms, detect chemicals dissolved in water, build awareness of their surroundings despite the darkness. Some Dumbo octopuses have been found with parasites, nematodes living in their digestive system, crustaceans attached to their skin. Even in the deepest ocean, nothing lives alone. Everything hosts something else. The lifespan of Dumbo octopuses is unknown. We have never tracked one long enough to measure it. They might live 3 years, maybe 5, possibly longer. Their slow metabolism suggests longer lifespans. But without data, we can only guess. What we know is that they represent a unique evolutionary path. Octopuses that left the seafloor, that evolved to swim instead of crawl, that adapted to the deepest parts of the ocean, They are proof that octopus body plans are flexible, that evolution can reshape familiar forms into something entirely new, and they share their habitat with creatures even stranger, fish that stand on the bottom instead of swimming. The tripod fish does not swim. It stands on the abyssal seafloor, motionless, sometimes for days. 3 fins extend from its body like legs, 2 pectoral fins, 1 elongated tail fin. Each fin is stiffened with rigid rays. Each one acts as a support column. The fish positions itself facing into the current. The fins plant in the sediment. The body lifts several inches above the bottom. The fish becomes a tripod. This stance serves a purpose. Lifting above the seafloor puts the fish in the current. Faster water flows above, the sediment. This water carries more food, more organic particles, more prey. The fish holds its pectoral fins forward, extended like antennae. These fins are covered in sensory cells. They detect vibration, chemical traces, anything moving in the current. When prey drifts past, the fish senses it, turns its head, opens its mouth. The prey gets sucked in. The fish returns to its waiting position. No chase, no pursuit. Just patience and precision. Tripod fish can hold this position for days, possibly weeks. They have been filmed by underwater cameras staying absolutely still for hours. The only movement is their gills opening and closing, breathing. This strategy requires almost no energy. The fins lock into position. The muscles barely work. The fish becomes a living statue. Food in the abyss is scarce. Energy must be conserved. Moving wastes calories. Hunting actively wastes calories. Standing still and waiting is the most efficient strategy. Different tripod fish species have different fin lengths. Some stand 6 inches tall, others over a foot. The height depends on where they live, on the characteristics of the local current. In areas with strong currents, shorter fins work better. Less leverage for the water to push against. More stability. In areas with weak currents, longer fins lift the fish higher. Get it into faster moving water above the boundary layer. Tripod fish are hermaphrodites. They have both male and female reproductive organs. This solves a major problem in the abyss. Finding a mate is nearly impossible. The ocean is vast. Population density is low. 2 fish might drift for their entire lives without encountering each other. But if you are both male and female, you do not need another fish. You can fertilize your own eggs. Self-fertilization is not ideal genetically. It reduces genetic diversity, but it is better than never reproducing at all. When conditions are favorable, tripod fish release both eggs and sperm into the water. Some eggs get fertilized by their own sperm. Some might get fertilized by sperm from other tripod fish, if any are nearby. The fertilized eggs drift with currents. The larvae develop in the water column. They feed on microscopic organisms. Eventually, they settle to the seafloor, transform into adults, begin their own waiting vigil. Tripod fish have extremely sensitive lateral lines. These detect pressure changes in the water, vibrations, movement. A shrimp kicking its tail 20 feet away creates ripples. The tripod fish detects them, knows something is there, knows the approximate direction and distance. If the shrimp drifts closer, the fish prepares, angles its mouth, waits for the perfect moment, strikes. The strike is not fast like a surface predator. It is deliberate, controlled. The mouth opens, creates negative pressure. Water rushes in. The prey comes with it. Tripod fish eyes are small, reduced. Vision is not critical at these depths. Chemoreception and mechanoreception are more important. Some tripod fish species are completely blind. Their eyes have degenerated to non-functional remnants. They navigate entirely through other senses. Their skin is dark, black or dark brown. This might reduce bioluminescent detection by predators. A dark fish against a dark background is harder to see even with bioluminescence. Tripod fish are found in all major oceans, wherever there is abyssal plain. They are one of the most successful deep-sea fish families, but successful does not mean common. Their population density is still low. You might survey a square mile of abyssal seafloor and find 3 tripod fish, maybe 5. They are specialists, adapted to a specific niche. That niche is vast. It covers millions of square miles. But within that vastness, each fish occupies a tiny territory, standing, waiting, surviving on patience and efficiency. And they are not alone down there. Another fish dominates the abyssal zone even more completely. Grenadier fish are everywhere. In the abyss, they might be the most abundant fish on Earth by biomass. More grenadiers exist than all salmon, all tuna, all cod, but most people have never heard of them. They have large heads, tapered bodies that end in a thin tail, no tail fin, just a pointed tip. They look like tadpoles that grew teeth and eyes. Their bodies are covered in scales, but these are not normal scales. They have sharp ridges, spines. Touch a grenadier and you might get cut. The scales create sound. When grenadiers swim, the scales rub together, produce clicks and rattles. They use this sound to communicate, to locate each other in darkness. Each species has a different acoustic signature, different rhythm, different frequency. They can identify their own kind by sound alone. Grenadiers cruise along the seafloor, swimming slowly, searching for food. They eat anything organic. Worms, shrimp, small fish, carrion, marine snow. They are opportunistic, not specialized. This flexibility is why they are so successful. When food is scarce, you cannot afford to be picky. Their mouths face downward, designed for bottom feeding. They root through sediment like pigs rooting through soil, sucking up whatever they find. Some species have barbels, whisker-like organs hanging from the chin. These are chemoreceptors. They detect dissolved chemicals, help the fish locate buried prey. Grenadiers have large eyes, bigger than you would expect for their body size. These eyes are adapted for detecting bioluminescence. They can see the faintest glow. But in total darkness, even excellent eyes have limits. So grenadiers also rely heavily on their lateral line system, on sound, on chemical sensing. They swim in loose aggregations, not schools exactly, but they stay within sensory range of each other. They communicate with scale sounds, with chemical signals. When one grenadier finds food, others nearby sense it. They converge, feed together. This increases individual feeding success, but it also creates competition, aggression. Grenadiers fight over carcasses, over concentrations of prey. They bite, ram each other, drive competitors away. Despite the abundance of grenadiers, predation on them is rare. Few predators exist at abyssal depths, and grenadiers are not easy prey. They are fast when threatened. Their spiny scales make them difficult to swallow. Most grenadier mortality comes from starvation, from old age, from disease, not from predation. Some grenadier species migrate vertically. They rise toward the surface at night to feed, descend to the abyss during the day. This behavior is common in younger grenadiers. Adults tend to stay deep. Reproduction varies by species. Some release eggs into the water column. The eggs drift and develop in the open ocean. Others lay eggs on the seafloor. Females produce thousands of eggs, tens of thousands. Fertilization is external. Males and females release gametes into the water simultaneously. Success rate is low, most eggs never get fertilized. Most larvae never survive to adulthood, but the sheer number of eggs compensates. Grenadiers grow slowly, take years to reach maturity, but once mature, they can reproduce multiple times. Some species might spawn every year for decades. Their flesh is watery, low in muscle protein, high in water content. This is typical of abyssal fish. It reduces density, provides neutral buoyancy, but it also makes them poor food for humans. Grenadier flesh falls apart when cooked, turns mushy, has a bland taste. They are sometimes called as bycatch. In deep-sea trawls targeting other species, the Grenadiers get discarded. They have no commercial value. This might be fortunate. If Grenadiers were commercially valuable, they would be overfished. Their slow growth and late maturity make them vulnerable to population collapse. As it is, they remain abundant. The dominant fish of the abyss. Swimming through the darkness in numbers we can barely comprehend. Every deep-sea camera deployment captures Grenadiers. Every net pulled from abyssal depths contains them. They are the background noise of the deep ocean. And among the creatures they encounter on the seafloor are some of the largest invertebrates ever recorded. Amphipods are crustaceans, related to shrimp. In shallow water, they grow to maybe an inch long. Beach hoppers, small scavengers that burrow in sand. In the Hadal trenches, they grow to 14 inches, the size of a dinner plate, with bodies thick and armored, legs like metal rods. This is the most extreme example of deep-sea gigantism, an organism that increases in size by more than 10 times its shallow water relatives. Scientists are not completely sure why. The theories for deep-sea gigantism all apply: cold water, high oxygen, long lifespan, low predation, but amphipods take it further than anything else. They are not just big, they are enormous. These giant amphipods are scavengers, opportunistic feeders. They eat anything organic that reaches the seafloor: dead fish, dead whales, wood, plant matter washed from land. When food arrives, they swarm, hundreds of them, covering the carcass like a living blanket. They eat aggressively, tearing off chunks of flesh, competing with each other. A dead fish dropped to the Hadal zone gets stripped to bones within hours. The amphipods are that efficient, that numerous. They have powerful mandibles, strong enough to crunch through bone. They process everything: scales, cartilage, organs. Nothing gets wasted. Between feeding events, they might wait months, maybe years. Their metabolism slows to near nothing. They drift through the water or crawl across the seafloor, conserving energy. Their exoskeleton is thick, reinforced. This provides protection from pressure, from predators, from the impacts of moving across sharp rocks. They have large eyes, compound eyes like insects. These eyes can detect bioluminescence, track glowing organisms, see in near total darkness. Their antennae are long, covered in chemoreceptors. They can smell food from miles away, detect the chemical signature of decomposition, follow it to the source. When researchers deploy baited cameras in trenches, amphipods appear within hours, sometimes minutes. They sense the bait, converge from all directions. The footage shows them crawling over each other, fighting for position. The largest amphipods dominate. They get the best feeding spots. The smaller ones wait for scraps. Some species of giant amphipods are predatory. They hunt smaller organisms, worms, other crustaceans, small fish. They ambush from the sediment, bury themselves, wait for prey to pass overhead, then lunge, grab with their legs, pull prey down into the mud. Their legs are lined with spines. These grip slippery prey, prevent escape. Once caught, the prey gets pulled to the mandibles, gets torn apart. Giant amphipods can survive extreme pressure, over 16,000 pounds per square inch in the deepest trenches. They contain the same pressure-resistant proteins as snailfish, TMAO in high concentrations. They also have flexible joints. Their exoskeleton can compress slightly under pressure. This prevents cracking, prevents structural failure. Reproduction in giant amphipods is poorly understood. We assume they mate like shallow water amphipods. The male transfers sperm to the female. She carries fertilized eggs in a brood pouch. The eggs develop over weeks or months. When ready, the juveniles are released. They look like miniature adults. They begin feeding immediately. Growth takes years. maybe decades. A 14-inch amphipod might be 30 years old, maybe older. Their lifespan is unknown. No one has tracked a giant amphipod long enough to measure it. They might live 50 years, possibly longer. What is known is that they are incredibly tough, resilient. They can survive conditions that would kill most other organisms. Experiments have brought giant amphipods to the surface. They survive for hours in aquarium tanks. They adjust to the pressure change, the temperature change, but they always die eventually. They are too specialized for trench life. Surface conditions are too different, too warm, too bright, too much oxygen. Giant amphipods represent an evolutionary endpoint. An organism that has adapted so completely to the Hadal environment that it cannot exist anywhere else. They are locked into the trenches, into the deepest places on Earth. And those places are about to get even more extreme. At 20,000 feet, you enter the Hadal zone, named after Hades, the Greek underworld, the realm of the dead. This is where ocean trenches begin. Places where one tectonic plate slides beneath another, where the seafloor tears open, where Earth's crust bends downward into canyons deeper than mountains at all. The Hadal zone is not continuous. It exists only in trenches, isolated pockets scattered across the ocean. The Mariana Trench, the Tonga Trench, the Kermadec Trench, the Philippine Trench. Each trench is an isolated ecosystem. Species evolve independently. What lives in one trench might not exist in another. Geographic isolation on a scale usually seen on islands. Pressure in the Hadal zone exceeds 15,000 pounds per square inch. This is the weight of a passenger jet pressing on every square inch of your body. Submarines cannot reach these depths. The holes would implode. Only specialized vehicles with ceramic pressure spheres can descend this far, and only a handful exist. Humans have visited the Hadal zone fewer than 20 times, total, in all of history. We have sent more people into space than into the deepest ocean. The temperature is just above freezing, stable, unchanging, has been the same for millions of years. The darkness is absolute. No bioluminescence from the water column above reaches this deep. The only light comes from organisms that live in the trenches themselves. Food is extremely scarce. Most organic matter gets consumed in the zones above. What reaches the Hadal zone is the final scraps. Particles so small they are barely nutritious, except for whale falls and woodfalls, large objects that sink fast enough to bypass the upper scavengers. When these land in a trench, they create oases, concentrations of life that persist for decades. The sediment in trenches is unique. It contains particles from the continents, washed into the ocean by rivers, carried by currents, settling slowly over thousands of miles. Some trenches contain sediment over a mile thick, layer upon layer of particles compacted under pressure. This sediment records geological history, climate changes, ocean chemistry shifts. Organisms that burrow into this sediment are processing the history of Earth, consuming the past, turning it into energy. The walls of trenches are steep, sometimes vertical. Organisms can cling to these walls, hide in crevices. Different species live on the walls than on the floor. This creates vertical zonation, layers of life stacked on top of each other, each adapted to specific conditions, specific pressure ranges, specific food sources. Trenches also experience earthquakes. The tectonic plates grinding against each other create tremors, landslides, sudden shifts in the seafloor. Organisms here must tolerate not just pressure and cold and darkness, they must tolerate geological instability. The ground literally moves beneath them. Some trenches have hydrothermal vents, cracks where superheated water escapes from beneath the crust. These vents support chemosynthetic ecosystems, life that does not depend on the sun. But most of the Hadal zone is cold, still, quiet. A world of sediment and darkness and crushing pressure. The species that live here are specialists. Snailfish adapted to pressure. Amphipods adapted to scavenging. Sea cucumbers adapted to sediment processing. Nothing from the surface can survive here. Nothing from the abyssal zone can survive here. The Hadal zone is its own world, and the deepest parts of that world are barely explored. We have sent cameras to a few trenches, collected samples from a few locations, but most of the Hadal zone remains unvisited. There could be species down there we cannot imagine, adaptations we have never seen, ecosystems operating on principles we do not understand. The Hadal zone represents the final frontier of ocean exploration, the last unknown territory on Earth, and we are only beginning to discover what lives in it. The Mariana snailfish was discovered in 2014, found at depths exceeding 26,000 feet. In the Mariana Trench, the deepest place on Earth. It should not exist. The pressure should prevent cellular function, should denature proteins, should make life impossible. But the snailfish swims, hunts, reproduces. It has solved every problem the Hadal zone presents. Its body is pale, almost translucent. You can see internal structures through the skin. The skull is partially visible. The vertebrae show through the flesh. This is not because the fish is unhealthy. This is adaptation. Reduced pigmentation saves energy. There is no light to hide from, no predators that hunt by sight. The snailfish has a large head, small eyes, a mouth that extends almost to the gill openings. When it feeds, the mouth becomes a funnel. It sucks in amphipod's hole. Its body is gelatinous, watery, almost no muscle density. This reduces metabolic needs, lets it survive on minimal food. The bones are soft, cartilaginous, lightly calcified. This gives flexibility, prevents fractures under pressure. Its stomach can expand dramatically. The fish can swallow prey nearly as large as itself. The stomach distends, the skin stretches, digestion takes days or weeks. Between meals, the fish might go months without eating. Its metabolism is so slow that a single amphipod provides energy for weeks. The snailfish uses TMAO to protect its proteins, concentrations higher than any other fish. This molecule stabilizes protein structure, prevents pressure from destroying enzymatic function. But TMAO has limits. Above certain concentrations, it becomes toxic, disrupts cellular processes. The Mariana snailfish is pushing that limit, operating at the maximum TMAO concentration biology can tolerate. This suggests a depth limit for fish, somewhere around 27,000 to 28,000 feet. Below that, even TMAO cannot protect proteins. Fish cannot exist. The Mariana snailfish is at the edge of that limit. It represents the deepest possible fish, the absolute maximum depth for vertebrate life. Scientists have deployed cameras deeper than 27,000 feet. They have not found fish, they found amphipods, sea cucumbers, invertebrates with fundamentally different biochemistry, but no fish. The snailfish is the end of the line. It reproduces like other snailfish. Females lay eggs on the seafloor. Males fertilize them. Development is slow. Months pass before the youngs hatch. When they do, they are miniature adults, fully formed, ready to hunt. They feed on smaller amphipods, gradually work up to larger prey as they grow. Growth takes years, maybe decades. A full-sized Mariana snailfish might be 20 or 30 years old. We do not know for certain. Their predators are unclear. The Hadal zone has few large predators. Maybe larger snailfish eat smaller ones. Maybe amphipods scavenge dead or dying fish. But active predation seems rare. Most mortality probably comes from starvation, from unsuccessful hunts, from the simple difficulty of surviving in the most extreme environment on Earth. The Mariana snailfish was not discovered by accident. Scientists specifically targeted the Mariana Trench. They deployed landers, baited cameras. They recorded hours of footage. The snailfish appeared in multiple videos, swimming near the bait, feeding on amphipods attracted to the bait, apparently common in its habitat. This was shocking. The deepest fish on Earth is not rare, not a lone survivor. It has a population, a breeding community. Other trenches might have their own snailfish species. The Kermadec Trench has snailfish at 24,000 feet. Different species, adapted to local conditions. Each trench's snailfish evolved independently. They are convergent evolution. Different lineages solving the same problems with similar solutions. All have gelatinous bodies. All have high TMAO. All have reduced skeletons. All occupy the same ecological niche. This suggests that the snailfish body plan is optimal for Hadal fish. Evolution discovered the same solution multiple times in different locations. The Mariana snailfish might be the most extreme fish on Earth, the one operating closest to absolute limits, but it is not struggling. It is thriving. In the deepest, darkest, most hostile environment our planet offers, this fish found a way. It exists where nothing should. It hunts where submarines implode, and it shares that environment with creatures even more alien.